Chapter 11, time to stimulate a muscle cell. So we're going to look at membrane potential as well as our action potential. So our muscle cells are electrically excitable. So when we have voltage changes, it's due to the stimulation of that muscle cell. There we go. Okay, so just some vocabulary for us. We have voltage, which is our electrical potential, the difference between one point and another. So if we're looking at the voltage between the inside and the outside of the cell, it's our resting membrane potential. So this resting membrane potential, which is about negative 90 millivolts in unstimulated skeletal muscle cells. So in other cells, it'll be different numbers. For example, in neurons, it's negative 70. This charge difference is maintained by our sodium-potassium pump. So the ones we talked about in Chapter 3 in our cell chapter, that sodium-potassium pump also works here. So let's look at some basic vocabulary. So an action potential is a quick change, a depolarization, and reset, repolarization in the charge difference, which is our membrane potential of a section of cell membrane. So a quick change or reset of that charge difference. So a quick depolarization and quick repolarization of our membrane potential. So our membrane potential, again, is the difference in charge between the inside and outside of cell. At rest, it is more negatively charged on the inside of the cell. So on the right side pictures are action potential, so our depolarization and repolarization, going back to our resting membrane potential. There we go. Okay, so in an unstimulated cell, if I took an electrodes and I place one on the outside, one on the inside of the cell, I have a recording of negative 90 millivolts. So what causes this difference? We have an excess of sodium ions on the outside, again, due to that sodium-potassium pump. And also because of that pump, we have more potassium ions on the inside. So this sodium-potassium pump, again, remembering that you have to use energy in order to set up this gradient. So you're going to have a sodium gradient and a potassium gradient that we're actually going to use for our action potential. There's also more negatively charged anions, so negatively charged molecules that are on the inside of the cell that make it more negative on the inside. For our stimulation, so we have a closed ligand-gated ion channel and a chemical signal. A ligand, it has to bind to this ion channel, so that ligand is the key that opens up the gate. So when that happens, that ligand bind, it causes a inflow of ions. So the ions are going to go down their concentration gradient depending on what type of ion channel it is. So you do have some ion channels that are for positively charged ions, such as sodium, and some that are for negatively charged ions, such as chloride. So it just depends on the actual channel. Now for muscle, we'll focus just on sodium because that's what we're actually needing for our muscle contraction. So depending on the charge is going to determine whether the membrane potential is going to become less negative and going towards more positive, or your membrane potential is going to become even more negative. So it's either going to go towards the positive or towards the negative. So if positively charged ions flow into the cell, then our ion channel is an excitatory one. So our stimulation is going to be excitatory, and you're more likely to start an action potential. So our negative resting membrane potential, and you're going to go towards a concept called threshold. And that's where you actually are going to trigger your action potentials. But you have to get away from this negative resting membrane potential to a, slightly lesser negative number. The opposite situation is inhibitory. So these is when negatively charged ions flow into the cell, it'll make the inside charge of the cell even more negative, going away from your threshold, and so makes it less likely to start an action potential. Okay, so for our muscle, we're just going to only deal with the excitatory type of stimulation. So we have our negative millivolt difference between the inside and outside of the cell. At threshold, it's still a negative number. So when we have our acetylcholine, which is the ligand, it's going to bind to the acetylcholine receptors, which are our ligand-gated ion channels. When that happens, you have a little bit of movement of sodium and potassium going down their concentration gradients, but it's enough to make the inside of the cell more positive. When that happens, you eventually reach threshold and start off your action potential. So our first step in our action potential is depolarization. So in this situation, sodium ions rush into the cell, so going down their concentration gradient, and it's going to make the inside of the cell more positive. So let's look at a recording. Depolarization, sodium rushes into the cell, and therefore making the inside of the cell more and more positive to the point you actually make the inside of the charge of the cell actually be positive. So it's going to cross that 0 millivolt number. Okay, opposite side of our action potential, our reset back toward our resting membrane potential. So eventually our sodium-gated ion channels are going to close. Our potassium ion channels are then going to open. When that happens, potassium goes down its concentration gradient. It's also repelled by all this positive charge that is inside the cell. So that also helps to move potassium out the cell, remembering that like charges are going to repel each other. So for our repolarization, this side of our action potential, potassium goes out of the cell. It makes the cell more negative because all this positive is leaving the cell. And eventually you're going to return back to your resting membrane potential. So this is your basic action potential. You do have two more types of action potentials that you'll learn for anatomy. You'll have one for your nervous tissue, so your neuron cells. Those have a similar type of action potential. They just have an extra piece added onto it. And then for AP2, you'll learn about action potentials for our pacemaker cells as well as our regular cardiomyocytes, which have an extra piece that's added to this basic action potential. All right, let's make a contraction. So we have our electrical stimulation from our neuron. It's going to cause the release of acetylcholine from the axon terminal at the end of the neuron. That acetylcholine goes down its concentration gradient across the synaptic cleft, binding to our acetylcholine receptors. So acetylcholine receptors, again, are sodium and potassium voltage-gated ion channels, and so it's going to cause them to move into the cell. Basically, it's going to be enough to trigger your muscle cell to start a action potential. So what we just talked about last time, or slightly earlier. So when this action potential is started, it travels along our sarcolemma, so our plasma membrane of the cell. It moves into a T-tubule, so part of our triad. When that action potential is in this T-tubule, it signals the release of calcium from our sarcoplasmic reticulum. That calcium goes down its concentration gradient to our myofilaments, particularly our thin ones, because that is where our regulatory proteins are located. So when calcium binds to troponin, our little yellow snowman here, it causes a conformational change. Tropomyosin moves off of the active sites on actin. That allows for our myosin head to be able to bind to our active sites. So in order to get the myosin head in the correct position for binding, a ATP molecule binds to that myosin head. When it goes through hydrolysis, so we're breaking that bond, releasing the energy, causing a conformational change that's going to put the myosin head in the correct position. So this powers up our movement for our contraction. So creates our recovery stroke, getting ready to be able to bind and pull on our thin filaments. So if you kind of think of it like a 6-shooter gun where you have the cocking mechanism on the back of the gun, just like that. So we cock it back so it's ready in position to actually fire. Okay, so we make a bridge between our myosin head and our actin filament. Okay, when ADP and the phosphate group releases from our myosin head goes back into its original position. Because it's bound to one of those actin subunits, it causes the contraction. So it's going to pull on our actin thin filament, causing the thin filament to slide inward and therefore causing the shortening of our sarcomere. To break this bridge between our myosin head and our actin subunit, another molecule ATP binds, and then you're starting to be ready for another contraction. So our ATP molecule will hydrolyze. We break that bond between the second and third phosphate group. You cock it back again so it's ready in position to have another contraction. Going back to our neuromuscular junction, our acetylcholine, it only stays on the receptor for a very short period of time. When acetylcholine releases, it's going to go down its concentration gradient and move away from the receptors. There's also your acetylcholine esterase enzyme to break down the acetylcholine into its acetyl group and the choline group. Those two pieces are taken up by your neuron, sit all the way back up to your neurosoma, your main cell body, so it can be put back together as acetylcholine. And then it's trafficked all the way back down to your axon terminal, packaged in a synaptic vesicle to be released again. Over in our triad, our calcium, it only binds for so long. Eventually the calcium releases. It's stored back in the terminal cisterns of our sarcoplasmic reticulum, so it can also be ready to be released for another contraction. Here, our myosin head, our ATP has hydrolyzed, our myosin, we're doing on this phase. We already talked about this, sorry. Okay, so our ADP, and phosphate group, after it's released, after you've had your actual pull, breaking of that bond using another ATP molecule, your calcium, it releases, tropomyosin binds to your active sites on actin again, so it blocks the ability for the myosin head to be able to bind to the active sites on actin. Even though there's YouTube videos that's going to cover this, I made a little mini one here. So we have our sarcomere, our Z-disc to Z-disc, our functional contractile unit of a muscle cell. So we have some overlap between our thick and thin filaments. These pieces being our myosin head sticking out there. So for a contraction, the thick filaments pull on the thin ones, causing even more overlap and a shortening of the sarcomere. So you have multiple sarcomere, they're going to shorten together to actually cause your contraction. Moving on to whole muscles, so looking at contraction, looking at multiple contractions closely put together, and different types of contraction. So with our muscle stimulation, not all stimulation actually triggers an actual contraction. So it has to be strong enough to actually recruit a motor unit in order to cause a contraction. So up top we have the different stimuli here, and we have different levels of stimulus, the red dashed line being our threshold. So this middle section, it's showing how many motor units are recruited, and then the bottom one is showing you the level of contraction, so seeing the actual shortening of our muscle. So going back to the top, our first two, they have not enough stimulation to reach threshold. No motor units are recruited, and so therefore you have no contraction. Number 3, that stimulus is at threshold. 1 motor unit is recruited, so you have a little contraction. So with the rest of these stimuli, they increase in stimulus voltage, so a stronger and stronger stimulus. So this recruits more motor units, so more motor units are contracting, therefore you're having an increase in your contraction strength up to a certain amount. So once all your motor units are recruited, so here at number 7, this is where we have our maximum contraction. So your contraction can't get any stronger because you have recruited all of your motor units to contract. This next part looks at the timing between the stimuli. So all of the stimuli in this section are the same strength. So here we have 3 different timing levels or frequency, different timing frequencies. So the far left, low frequency, middle is higher frequency, and the far right is a unnaturally high frequency. So for a low frequency stimulation, the time difference between our stimuli is long enough to allow for our muscle to fully relax. And so the strength of our muscle twitches is going to be the same. So you're starting from baseline every single time. The next one is a higher frequency timing of our stimulation. So in this case, you only have partial relaxation between muscle twitches. So you're not having to start all the way down from baseline. So you're building in strength on your contractions. So this is called temporal wave summation. So temporal relating to time. So in this case, the short time between our, what is that? There you go. Short time between our muscle twitches allows for the contraction strength to build on top of each other. Third one is the unnaturally high frequency. So in this case, this is more something done in like a lab setting where you have some partial relaxation until you get to the point of what's called a fused tetanus. So this is a fused contraction. There's absolutely no relaxation in between the twitches. You just have a smooth, steady contraction. So for our ATP energy sources for muscle, we have oxygen and glucose. For our muscle stimulation, let me see here. The most efficient way to make ATP is using aerobic respiration. So in order to do that, we need oxygen and some type of organic energy source. Muscles have their own storage for that, so we have myoglobin for oxygen, glycogen for our glucose. Even though muscles can use both pathways for making ATP, aerobic respiration is the more efficient one. Moving on to classes of muscle fibers. So I'm going to go over this really quickly. Basically, you have 2 different types. You have slow twitch, which are fatigue resistant. They're more your posture muscles. For example, keeping you sitting upright. So they have very slow contractions, very sustained movement. So very small motor units, so everything is very precise. So just making those slight adjustments for keeping your posture upright or holding you in a certain position. The other type are your fast twitch. So these are for more strong, quick movements. For example, your eye being a quick movement type of, very quick type of movement. You do have larger motor units, so you have all of these neurons firing at the same time, so you can have that quick or powerful movement type of situation. Another area would be your calf muscles and biceps brachii, so very strong movement as you are lifting something, or gastrocnemius, so your calf like trying to run away. So we've already covered this in chapter 5 for your differences between your different types of muscle tissue. So I'm actually going to skip through some of this, knowing that your striated, your cardiac muscle and your skeleton are both striated, and those kind of things. So the little simple comparisons between them. One thing about your, trying to find this, cardiac muscle is that it repairs using fibrosis instead of using regeneration. So skeletal muscle will regenerate, cardiac will not. It just becomes a scar tissue. Another one of your comparisons, so talking about them, they're slower in smooth muscle for your contractions because that's all internal organs. So you don't need stuff quickly moving. For example, like your stomach, that's smooth muscle. It's going to slowly contract and help you with your digestion, slow movement through your intestines, so you don't need to move too fast. One thing with your smooth muscle that's also different from your skeletal is instead of having a neuromuscular junction, you have what's called varicosities. So it kind of works like your sprayer. If you're country enough, you've seen these. They're water sprayers that are over a field, and at certain points, they'll release water onto your crop. So very similar is that each of these little bulbs is like a axon terminal. You're releasing your neurotransmitter, and it just kind of sprays over the general area. Remembering that your smooth muscle isn't going to be a precise movement. It's just going to be a slow, contained movement in a general area. Another difference between your, for example, smooth and skeletal muscle system is that your calcium binds to calmodulin. So instead of binding to troponin, the calcium binds to calmodulin. So just a different protein. It's still going to eventually trigger your contraction, except for in your smooth muscle, it's just going to be a lot slower because it's just a different type of function for that type of muscle. So that's it for chapter 11. So I'll talk to you later.